Okay, what if I told you the secret to building tiny robots, nanobots, that could deliver medicine right to a single cell wasn't just biology, but math? Yeah, advanced mathematics. Let's dive right in, because we're about to see how researchers are literally using pure math to write the instruction manual for the future of nanotechnology. And they're building it from the code of life itself. So when you see a molecule like this, you're probably thinking genetics, right? But scientists are looking at this in a whole new way. They see it as a construction material. This is what's called a K-armed branched DNA molecule. And instead of just reading its code, they're using its physical shape, its kind of sticky ends, as a microscopic Lego brick that can be programmed to build things all by itself. And what are they trying to build? Well, the potential is just incredible. This quote from the research paper gets right to the point. The main goal here is to get these DNA molecules to automatically assemble into tiny cages. We're talking just a few billionths of a meter wide that can carry life-saving drugs directly to, say, a cancer cell without touching any of the healthy cells around it. Yeah, this whole field, DNA nanotechnology, it promises to create these molecular machines that could completely revolutionize medicine. But to actually build them, we're up against a massive challenge. See, here's the heart of the problem. You can't exactly use a tiny pair of tweezers to snap these pieces together. It's not possible. You have to design them so perfectly that when you just mix millions of them in a solution, they build the exact complex structure you want, all on their own. It has to be a flawless self-assembly every single time. So what happens if that microscopic blueprint, the design of these little DNA bricks, has even a tiny mistake? And this slide just shows you the risk so clearly. On the left, you've got the target. That's the perfect, complete shape you want. But on the right, well, that's what happens with a bad blueprint. You get a bunch of useless fragments. Or even worse, you get completely different shapes. And if you're trying to build a drug delivery cage, close enough is a total failure. So the main takeaway here is this. The problem isn't the raw material. We have the DNA. The real challenge is having the perfect set of instructions to guarantee you get the right result every single time. And this is where the story gets really, really interesting. The solution comes from a field of pure math you might remember from school, graph theory. The study of dots and lines, or vertices and edges. Scientists figured out they could model this entire biological process as a mathematical graph. And that graph becomes the ultimate instruction manual. They call it the flexible tile model. It's a really brilliant way of translating all the complicated chemistry into simple mathematical roles. Just think of each DNA molecule as a tile, and the chemical bonds that link them up as bond edges. It basically turns biology into a puzzle. Let's break that down real quick. A tile is just our DNA building block. That's the vertex, the dot in our graph. And a bond edge, that's the unique chemical connection. Think of it like a smart glue that only lets specific tiles connect to each other. That's our edge, the line. The whole game is to figure out the minimum number of unique tiles and unique glues you need to get the job done. So within this model, you've got three possible outcomes. Scenario 1 is total chaos. Anything and everything can form. Scenario 2 is a little better. It at least stops smaller junk pieces from forming. But Scenario 3, that's the gold standard. That's where you guarantee that only the single exact structure you want can possibly form, nothing else. For medicine, you absolutely need Scenario 3. And this brings us right to the mission of the research we're looking at. These scientists set out to find the absolute bare minimum number of unique DNA tiles and bond edges needed to force a perfect Scenario 3 build for some really important complex shapes, because efficiency is everything. Okay, so how did they actually put this blueprint to the test? Well, they didn't just model simple stuff like cubes. They went after a family of incredibly complex shapes that have huge medical potential. The test subjects were Archimedean graphs. These are basically the skeletons of these really beautiful, symmetrical, but highly complex shapes. And because of that structure, they are the perfect candidates for those medical nanocages we've been talking about. But that same complexity also makes them incredibly hard to build correctly. So, a perfect test case. To solve this puzzle, they combined some deep graph theory, like identifying specific properties of these shapes, with modern computing. They actually wrote Python scripts to check their work and prove that no smaller or incorrect structures could possibly form. It was this amazing fusion of pure theory and computational power. And this is where it all comes together. After all that abstract math, what does it actually mean for the real world? Well, it means a revolution in how we do this, a revolution in efficiency. To see just how powerful this is, let's just focus on one of the results. We're going to look at a shape called the cubooctahedron. This table just lays it all out. It's fantastic. You see it right there. To build a perfect cuboctahedron, which has 12 of those DNA tiles, you need a minimum of exactly 8 unique bond edges, 8 types of that smart glue. 8, that's the magic number. It's not 7, it's not 9, it's exactly 8. Before this work, a lab might have to just guess, maybe trying dozens of different bond types, wasting so much time and material. But now, math gives us the precise, most efficient number possible.
This number is basically an answer key. It's a recipe that tells a scientist in a lab the most efficient way to design their DNA molecules. It completely takes the guesswork out of the process, and that saves a huge amount of time, money, and precious materials.
And by making this whole process predictable and super efficient, this mathematical blueprint, well, it brings those incredible medical applications, those targeted nanocages for fighting disease, one giant step closer to being a real-world therapy. So we're really at this amazing moment where abstract math is providing a direct, practical blueprint for building things in the physical world, but at the nanoscale. The math has given us the plan. The only question left is, what are we going to build with it?